Hello and welcome to IB Bio 1.3 notes. We'll be discussing cell membranes in this section. So first of all, the main idea is that the structure of biological membranes makes them fluid and dynamic. These are all just background information, context. You could look at it if you want, but you don't really have to memorize any of it. So the fluid mosaic model, we're going to talk about that first, and it's a model made in 1972 of the structure of the cell membrane. So it consists of a phospholipid bilayer, as we see here, with proteins, these blue objects embedded in the bilayer, making it look like a mosaic. So phospholipids are lipids consisting of one glycerol, two fatty acids, and one phosphate molecule. So we have this hydrophilic head right here, and this is where the glycerol and the phosphate molecule is. And then we have the two fatty acid tails, which are hydrophobic. So phospholipid bilayer has two layers of phospholipids that are arranged according to their properties. They're held together by weak hydrophobic bonds between the fatty acid tails. And these bonds can be broken and reformed, which gives the membrane its fluidity and flexibility. So saturated fatty acid tails are viscous, meaning they are rigid, while the unsaturated ones are fluid. So we said this before, phosphate heads are hydrophilic, while the fatty acids are hydrophobic. This makes it an amphipathic molecule, which is a molecule that has both hydrophilic and hydrophobic properties. So the hydrophilic head, phosphate head, faces the inside and the outside of the cell, since there's watery environment, while the tails face the inside where there's no water, since they're hydrophobic. Okay, now membrane proteins. So membrane proteins are a group of proteins with diverse structures. They're located in the membrane and are either integral proteins or peripheral proteins. And they support the plasma membrane in carrying out different functions. So first we'll start off with integral proteins, which are also amphipathic. They're both hydrophilic and hydrophobic, and they mostly pass completely through the membrane. So we have this, and these are all integral proteins. Then we have peripheral proteins, which are polar, meaning they're hydrophilic or have a charge, and they attach to the outside of the membrane only. So for example, here, these are both peripheral proteins. Then we also have some proteins have a channel, so channel proteins that allow the passive transport, meaning transport without energy of substances in and out of the cell. So this is an example of a channel protein. The substances just move straight through. Then we have a carrier protein, which is a protein that binds to substances on one side. So say it binds right here, and then the protein changes shape to transport them to the other side, or from here to here. And then protein pumps are also carrier proteins, but they use energy. Then we have recognition proteins, which are proteins that help in distinguishing self and non-self cells, meaning they help distinguish cells from one another, which is really important for the immune system. Then we have receptor proteins, which just help in the communication of information inside or outside of the cell. Finally, we have enzymes, which are a type of protein that help increase the rate of metabolic reactions within the cell. All right, glycolipids, these aren't proteins. These are a combination of phospholipids and carbohydrates. We see that right here. And they help maintain the structure of the membrane and also in cells differentiating between self and non-self cells. So when you draw the fluid mosaic model, you have to include the following. So you have to include individual phospholipid molecules. So these, they're shown by using the symbol of a circle for the hydrophilic head and two parallel lines for the hydrophobic tails. You need a range of membrane proteins, including integral and peripheral. So these are integral proteins, peripheral proteins, also integral. You have to include the following labels. So the phospholipid bilayer, which is this whole set. We have glycoprotein right here. This protein, we have glycolipids, and then integral and peripheral proteins, as well as cholesterol inside the fatty acid tails embedded in the membrane. We'll talk about that right now. Okay, so cholesterol is a steroid only in animal cells, and it is made up of a nonpolar, so a hydrophobic part, comprising of four ring structures, so one, two, three, four, a hydrocarbon tail right here, and a polar hydroxyl group, which is hydrophilic. So, since cholesterol is amphipathic, it can insert itself into cell membranes by interacting with the phospholipids. And now it is really vital to fluidity and permeability of the membrane to some solutes. So, for example, it restricts the movements of phospholipids and other molecules in warm temperatures, reducing membrane fluidity. However, at lower temperatures, it disrupts the regular packing of the hydrocarbon tails, which prevents the solidification of the membrane and allows the membrane to stay more fluid at lower temperatures. And this allows the membrane to function properly, and it also reduces membrane permeability to hydrophilic molecules and ions such as sodium and hydrogen. So you're going to need to know how temperature affects membrane fluidity through cholesterol. And yeah. Then we have the Davison-Danieli membrane model, which is an earlier model proposed in 1935. So it suggested that the ratio of the total lipid surface area to total cell membrane surface area was 2 to 1. It also suggested that the lipid bilayer was just completely covered with protein on both sides. So this model explained the membrane's effectiveness as a barrier to the movement of certain substances. However, it didn't explain how different types of membranes can carry out different functions, since it assumed all membranes are the same with the same proteins. Um, yeah, and most proteins are hydrophobic, which makes it unlikely to be facing the outer and inner part of the cell, which both contain watery environments. And then freeze fracturing revealed a rough, irregular surface inside the phospholipid bilayer, which means there could be no smooth protein on the outside. Now, freeze fracturing is just a method, a process in which the membrane was frozen and then cracked, which just revealed the phospholipid bilayer and membrane proteins. So later, the fluid mosaic model replaced this model by having separate proteins embedded in it, allowing the hydrophilic proteins and portions of proteins and phospholipids to be exposed to water, and the hydrophobic parts not to. Yeah, that's it for 1.3. Thank you.